Hello everyone, and welcome to this podcast for cell biology. In this podcast, we're going to talk about nucleic acids. This is one of the 4 macromolecules that we will talk about in cell biology. In this podcast, I'm going to talk about a few things. I'm going to start with a brief introduction to what the central dogma is. It's not mentioned in your book at this point, but it's a good background on why nucleic acids are important. Then we'll talk about what is a nucleic acid, the parts of a nucleic acid, what is it that makes a nucleic acid a nucleic acid. Then we'll talk about the 2 types of nucleic acids, first DNA and then RNA. Alright, so let's go ahead and get started with talking about the central dogma. You're going to learn a lot more about the central dogma and nucleic acids in general when you take genetics. But for now, let's think about the central dogma in its simplest form, and that's when you take DNA. It is then converted into RNA. The process where this occurs is called transcription. And then RNA is converted into proteins. And this process where RNA is made into proteins is called translation. The proteins, as we just talked about in the past several podcasts, is what provides the traits of an organism. It provides those catalytic activities, the structure, the defense, the transport. All those functions of proteins occur because we take DNA to RNA to proteins. The reason I like to talk about the central dogma at the beginning of the discussion of nucleic acids is because this process of going from DNA to proteins includes 2 of our nucleic acids, DNA and RNA. So together, these 2 make up our nucleic acids. The DNA, as we'll learn, is the hereditary unit. It's what is passed on from generation to generation. And the RNA is used to take that information from DNA and turn it into proteins. So they're critical molecules in our cell. So let's now take a closer look at the generic nucleic acid. What is a nucleic acid? What is it made of? And how does it look? Though we'll come back to DNA later, I'm just going to draw a double helix here. This double helix of DNA is composed of linked smaller molecules called nucleotides. These interlinking molecules of nucleotides are known as the functional unit of nucleic acids. The functional unit of DNA or RNA. You could also call them the monomer of nucleic acids. Now, nucleotides are made up of 3 structures: a sugar, a phosphate group, and a nitrogenous base. Whether you're talking about a nucleotide that will be part of RNA or part of DNA, it will have these 3 structures. Now, there will be some differences as we'll highlight in just a moment. But let's draw a simple nucleotide. We first start with our sugar, and attached to this sugar is a phosphate group. Also attached to this sugar is our nitrogenous base. Now, this sugar is different depending on if you're talking about RNA or DNA. So this sugar here is different depending on if you're talking about a nucleotide found in DNA or a nucleotide found in RNA. If it's a sugar found in DNA, it will be called deoxyribose. If it's a sugar found in RNA, it will be called ribose. They look very similar, but there's one very important difference. And that is at this position right here. Oh, I should also include, there's a carbon that comes off of here. So this position here, in ribose, there's a hydroxyl group. And in deoxyribose, there's just a hydrogen. I should also let you know that there are hydroxyl groups at each of these carbons. When it forms a nucleotide, this hydroxyl group at this position is lost because the phosphate group comes in there. But I want you to focus on this hydroxyl group here and this hydrogen here. This position on both molecules we call the 2' position. So we say ribose has a 2' hydroxyl group and deoxyribose has a 2' hydrogen. Seems like a small difference, and structurally it is a small difference, but it is what accounts for DNA being much more robust than RNA. But it is enough of a difference that allows DNA to be a longer lasting molecule than RNA. And we'll talk later why that makes RNA less stable. Another difference between the nucleotide found in DNA and RNA is found with the nitrogenous bases. And before I talk about that difference, I want to write down the 5 bases that are associated with DNA and RNA. They include 3 pyrimidines and 2 purines. The differences between pyrimidines and purines are mainly that pyrimidines are composed of a single ring and purines are composed of a double ring. If on an exam you're asked to draw a nucleotide, I'm not going to expect you to draw the detailed structure of a nucleotide that you can find in your book. I would just want you to indicate that pyrimidines are 1 ring, and you can just draw it like that, and purines are double rings. And I would just draw them connected like so. The way I always remember that pyrimidines contain 1 ring and purines contain 2 rings is that pyrimidines is a longer word, yet it only has 1 ring, and purines is a shorter word, but it has 2 rings. So it's kind of counterintuitive. So that's always helped me remember that. Pyrimidines, there are 3 of them that you need to know. The first is cytosine. Often we just abbreviate that as a C. Uracil, we often just call that U. And thymine, which we just often call T. The way I always remember that these 3 are pyrimidines is that it spells the word cut, C-U-T. And I know this is going to sound goofy, but this is how I've always remembered them. If you think of a pyrimidine, I think of a pyramid. And if I'm thinking of a pyramid structure, I'm thinking that the very top of that pyramid, it would be sharp, and it could cut my finger. So C-U-T for cutting my finger on the top of a pyramid. All right, I know it's goofy, but it's always worked for me, and I've always remembered it that way. Purines, there are 2 purines you need to know. Guanine and adenine. Guanines are often just abbreviated as G and adenine as A. So the difference between DNA and RNA in regards to nitrogenous bases is that DNA contains these 4 bases: cytosine, thymine, guanine, and adenine. And RNA contains cytosine, uracil, guanine, and adenine. So you can see that the difference is that DNA contains the pyrimidine thymine, and RNA contains the pyrimidine uracil. So those are the differences between the nucleotides found in RNA or DNA. The phosphate remains the same in both of them. Now I want to just spend a moment talking about how these nucleotides come together. Okay, so let's now talk about how we're going to string these nucleotides together to form the nucleic acid. And let's begin by just drawing 1 nucleotide. And I'm just going to abbreviate my base here with this circle. And I'm going to make this DNA, so I'm not going to put a hydroxyl group here. If nothing's written there, you can assume it's just a hydrogen. Remember, we called that the 2' position. This position where this hydroxyl group is coming off, we call the 3'. So we call this the 3' hydroxyl group. Up here, this is the 5' carbon, and attached to the 5' carbon is the phosphate group. So we call this the 5' phosphate. As a nucleotide is being added to this original nucleotide, it will be added to this 3' hydroxyl group. And as this phosphate group interacts with this hydroxyl group, they form a bond with the phosphate and the 4 oxygens. And then we have our 3' hydroxyl group at this end. And then we can add another nucleotide, just like so. And we'll keep adding nucleotides until there are no more nucleotides to add. But a couple things I want to point out to you as you look at this drawing here is that the nucleic acid sequence of these nucleotides always starts with the 5' phosphate group and ends with the 3' hydroxyl group. The bases are always stuck into the middle here, and these bases can alternate in any different order. Ultimately, as you'll learn later, this is what's going to help determine which protein is going to be produced. But as I wrote these here, we know that this is a pyrimidine, this is a purine, and this is a pyrimidine. We don't know which ones they are, but we do know that they're pyrimidines and purines based on how I drew the rings. And since we know it's DNA, we know that the pyrimidines can't be uracil. And the last thing I want to say here before we move on is what we call this bond that brings those two nucleotides together. We call that the phosphodiester bond. It's a covalent bond, but we give it a unique name, calling it a phosphodiester bond. Okay, let's move on to talking about DNA structure. Just like when we talked about protein structure, DNA also has different levels. So the primary structure of DNA is similar to what you might think of if you think about the primary structure of protein. It is just the linear sequence of bases. We could draw the structure, we're not. The way we usually indicate the primary structure of DNA is by writing 5' and then the sequence of the nucleotides. So it might be G, C, A, T, T, A, C, C, A, G, A, T, and any other sequences that might come through. But just to end it here, I'm going to say this is our primary structure of DNA. Again, it can be in any sequence, and ultimately this sequence will determine which kinds of proteins will be made. And just like protein was characterized by peptide bonds, the bond that characterizes the primary structure of DNA is what we just talked about on the last slide. It would be these repeating phosphodiester bonds. So now let's think about the secondary structure of DNA. The secondary structure of DNA is the double helix. And this forms when we have two strands of DNA. I'm going to draw them vertically now, and not horizontally like I had up there, but vertically, because I wanted to point a few things out. And I think it's easier to look at it in this orientation. Remember, we said they're always written 5' to 3'. However, the double helix has a unique requirement, and that is that it is anti-parallel, which means that the other strand is written 5' to 3', but upside down. So this strand here, 5' phosphate, with all the different nucleotides to the end of the 3' hydroxyl group. Here it's written upside down from the 5' phosphate to the 3' hydroxyl group. It has to be arranged that way in order to get the double helix that we see. The double helix is characterized by the bases of one nucleotide. So this is one nucleotide here, this is another nucleotide here. We always have a pyrimidine binding to a purine, or a pyrimidine on this strand binding to a purine on this strand. But it's always one on one strand, a pyrimidine on one strand, and a purine on the other strand. Which strand has which, it doesn't matter so much. And the reason we need that is so that they can fit into this double helix. If you had two purines, they would get, there wouldn't be enough room there. So just like in proteins, the secondary structures were held together by hydrogen bonds, same thing here. These bases of the two alternating bases here are held together by hydrogen bonds. So let me write that down here. Bases of opposite strands held together by hydrogen bonds. Now I'm going to complicate it just a little bit more. I said that it had to be a purine binding to a pyrimidine, and that is true, but there are certain rules that must be applied here. So for instance, G's or guanines only bind to cytosines. So the purine guanine binds to the pyrimidine cytosine. It doesn't bind to thymine. So the other two bases that are left, the purine adenine will only bind to the pyrimidine thymine. I remember these because G's and C's, the letters that is, have curves to them. So we have a curve here for C and a curve here for G. And that's always helped me remember that those two bind to each other. A's and T's I remember because they can be drawn with just straight lines as opposed to curves. You may have noticed that I drew 3 lines, 3 hydrogen bonds between guanine and cytosine here. The reason I did that is because guanines and cytosines are held together by 3 hydrogen bonds. And adenines and thymines, I drew 2 lines here because they're held together by 2 hydrogen bonds. So, if on this strand here, I drew the following bases, let's say I drew a G, a C, a T, and an A. You should be able to predict what's going to be on this strand over here, because G's will always bind to C's, C's will always bind to G's, T's will always bind to A's, and A's will always bind to T's. If I ask you to draw this, I ran out of room here, but I would want you to indicate the 3 hydrogen bonds for G's and C's, and the 2 for T's and A's. I always remember 3 for G's and C's because 3 rhymes with G's and C's. Now, we call this the secondary structure, and I said it was a double helix, so it doesn't normally look like this in its secondary structure form. Rather, it forms a helix like so, where you still have your 5' and your 3', and it's still anti-parallel. So that would be the secondary structure. But if you're asked to draw base pairs like this, it's easier to draw it in this ladder formation as opposed to something like this. Alright, lastly, let's talk about DNA's tertiary structure. So the tertiary structure of DNA is analogous to the protein's tertiary structure in the sense that in DNA we will take the double helix and it will be compacted to form a very tight structure. And so here you may have a DNA molecule like this, and it's going to be compacted to form a very organized structure called a chromosome. You'll learn more about chromosomes in genetics, but all I want you to know now is that the tertiary structure of DNA is the chromosome. Okay, before we move on, I want to mention 4 scientists that were pivotal in understanding the structure of DNA. The first I'm going to mention is a scientist by the name of Chargaff. And I want to mention him because he's the one that established those rules we've already talked about, that A's bind to T's and G's bind to C's. And though I didn't mention it before, but will when we talk about RNA, A's also bind to U's or uracils. So he's really important in making this discovery. Okay, the next 3 scientists I want to talk about had a much more direct role in solving the structure of DNA. The first scientist is Rosalind Franklin. The next 2 are Francis Crick and James Watson. The story of Rosalind Franklin, Francis Crick, and James Watson is a fascinating story of how science is done and how sometimes people behave unethically in science. We won't have time to talk about that, at least not in this podcast, but maybe at some other point we will. But what I do want you to know is that Rosalind Franklin was an amazing X-ray crystallographer. What she could do is take X-rays and shoot them at crystal structures of various molecules, and then was able to use those patterns of the way the X-rays bounced off the crystals to solve the 3-dimensional structure of those molecules. And of course, the reason why I'm talking about her today is because she was the one that was able to get the best patterns of DNA, which led to solving the structure of DNA. Now, Francis Crick and James Watson, I'm not trying to minimize what they did because they were both very, very smart individuals, but they were model builders. And I don't mean that as a bad thing. That's just what they did. They were able to take data that had previously been published and data that they suspiciously got from Rosalind Franklin, and they were able to piece those data together in such a way that they were able to determine what the structure of DNA was. They immediately recognized when they looked at the structure of DNA that it had the properties necessary to be the molecule of inheritance. Further experimentation went on to show that indeed DNA held the material, held the information necessary for life to exist and for life to continue to exist. Their work that led to discovering the structure of DNA with data from Rosalind Franklin and other data that happened to have been published led to these two individuals, Francis Crick and James Watson, as well as a third individual, Maurice Wilkins, whom I didn't mention here. They all won the Nobel Prize. Rosalind Franklin, however, did not win the Nobel Prize. Maybe in class we can talk about why she didn't win the Nobel Prize. Okay, now I want to talk about RNA structure. RNA has a primary structure, and it's just like the DNA primary structure. It's written 5' to 3' with the interlinking nucleotides written in the direction of 5' to 3'. They're also held together by phosphodiester bonds. Next, let's talk about RNA's secondary structure. Now, it doesn't form the double helix like we saw with DNA, but it can have base pairing. So for instance, these sequences, say right here in the primary structure, if they're complementary, meaning they are match up with guanines being able to bind to cytosines and adenines being able to bind to uracils, then they'll form what we call stem loop structures, where on one side you might have an adenine, and over here you'd have a uracil, you might have a guanine, a cytosine, something like this. Still using the same base pair patterns and still with the same number of hydrogen bonds between the two. It just doesn't form that double helix. So now I want to just say a couple things about RNA function. When you take genetics, we'll go into great detail. There are many different kinds of RNA. You've probably heard about a few of these from other classes, maybe from high school, but you'll learn that the family of RNA molecules is a very diverse family. But their main function, almost all of them play some kind of role in protein synthesis or gene regulation, which is linked to protein synthesis. But so these two functions here, protein synthesis and gene regulation, falls largely into the area of RNA and what they do in a cell. I mentioned this at the end of the protein podcast, how RNA is a leading candidate as that first molecule of life. And one of the reasons why, and it's appropriate to mention here, is that RNA can function as something called a ribozyme. As the name indicates, it's an RNA molecule that functions as an enzyme. Though most catalytic molecules in a cell use proteins, there are a few steps in protein synthesis that rely on RNA as the catalytic molecule. And when they act as catalytic molecules, we call them ribozymes. So make sure you remember that ribozymes are RNA molecules that function as enzymes. So that's all I have for this podcast. So let's close with a quick summary. We began by talking about the central dogma to introduce the importance of nucleic acids. We then talked about how nucleotides are the functional units of nucleic acids. So together, a string of nucleotides make nucleic acids. We talked about the different properties of nucleotides, how they differ between RNA and DNA, what makes up a nucleotide, which led us to the two nucleic acids, DNA and RNA. With these, we talked about the structure, we talked about base pairing rules, we talked about the orientation that we write them, and a few other things that you'll recall. And we also talked about some famous scientists. We only mentioned 4 of them, so make sure you know them, and what their role was in solving the structure of DNA. Okay, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.